and we'll turn the music on.
Yeah.
From Microbe TV, this is TWiEVO, This Week in Evolution, episode 109, recorded on December 19th, 2024. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from a bright Salt Lake City, Nels LD.
Yeah. Hey, Vincent. Good to see you. Great to be here. Thank you, everyone, for your flexibility. We're live streaming at a different time than usual. And Vincent, you and I were so caught up in the pre-show chitter chatter. We're a minute or two
Late.
Yeah.
And then all of my graphics were not here, so I had to get those up. But it's okay. You can't change these things. That's the problem. Anyway, nice to see you again. Welcome everybody on this is the ultimate episode of TWiEVO for 2024, right?
Yeah, big year for TWiEVO. Year in review, and our special holiday bonus edition where we first talk about cats. Can't resist for the holidays. But yeah, Vincent, we celebrated our 100th episode of TWiEVO together.
Yeah, back in March, right?
We were in vivo back in March.
Yeah,
at the Science Mill down in Johnson City, Texas.
That's right. That was fun.
Really fun. Great
fun
to be together, and had some fans show up who were in the neighborhood for South by Southwest, and some good friends. Rich Condit was on hand, and others. We enjoyed the hospitality of the Science Mill as well. So that was pretty memorable.
Thanks for joining us, moderators. Today we have Barb Mack, we have Tom Steinberg, and looks like it for now. Thanks for coming and moderating.
Yeah.
Tell us where you're from, folks. We'll go through here and say hello.
We'll say hello, and we'll extend our holiday greetings around the globe. Obviously, it's because we're live streaming a bit late in the day, we might have lost a few of our regulars who are hopefully sleeping. It's this is all being recorded, so if you aren't catching it live, no sweat. Catch up on the recording, and then join us for a live stream in the new year, 2025.
Barb Mack says, I'm looking forward to this talk because cats and science. Yeah. Let me move this down a bit here. I'm missing some of our graphics, but it's okay.
That's okay.
So I have it all set up nicely, and then the program updates, and boom, it's all gone.
Yep. Rebuild from scratch.
Jan is in sunny San Diego, where it's 19°C.
Yeah. Sounds great. Yeah, welcome.
Jason is in France. Cool. This is very cool. I think MK, I think, is from Eastern Massachusetts, if I'm not mistaken. Gary is in Colorado. Happy to be here in the TWiEVO oasis of sanity. Here on Microbe TV, in fact, you will get oases of science sanity, right? Because the rest of the country seems to be going science crazy in a bad way. And we've talked about this a little bit. But if you want your science explained, we're going to do it for you here. And for that, we need your support, microbe.tv/contribute. We're doing fundraising till the end of the year, so please go over there.
Oh, yeah.
Make a contribution. We'd really appreciate that.
Absolutely. Vincent, I think I saw a clip or two. You're as part of the fundraising efforts this year. A great idea to have some listeners record videos. And I think I saw one or two that were really fantastic, really inspiring.
Yeah, we had a bunch of listeners send in, and I've been posting their videos slowly. And I think we really appreciate that because they tell us why they like us. In fact, Tom, one of our moderators, sent in a little clip himself. Also here, another mod, Les is here. Welcome. Thank you for coming. Abdulaziz is from the Kingdom of Saudi Arabia.
Good to have you along. Good to see you again.
Welcome back.
Yeah.
Peter is in London.
London calling.
I like the international flavor here. Lise is in Columbus, Ohio.
Wonderful.
Yes, MK is in Eastern Massachusetts. I got that right. Pete is in London.
Wonderful. Got some night owls.
Abelure. I used to drink Abelure or Abelauer. I don't know how to say it. It's a good one. It's a good. Is it my favorite? I don't remember what my favorite is. I used to like Lafroig. It's really peaty.
Oh yeah.
There's some others that are not as peaty, but good. I can't remember. Macallan is very nice too.
Mm-hmm. Agreed. Good to stay warm on a holiday evening.
Elizabeth is in West Virginia.
Welcome.
Barb Max says the whole world's mad right now, so this is a great place to hang out. Yeah, it's science mad. They're crazy. I mean, yeah, you heard that RFK wants to stop the polio vaccine. It's just ridiculous. Just being stupid for no good reason, just to get attention. Don't you think?
I think like we're careening well off the rails, and it feels like, or the beginning of that, sort of back into a program of chaos. It feels like we're sort of, looking at the news headlines today. Sounds like the government might not be open by this weekend, which is sort of.
Why is that? What's happening with is money? They're running out of money again?
Well, no. So it's not the debt ceiling, although that's interestingly been thrown in as a curveball right at the end. But so the the continuing resolution, so just the budget appropriations, which have been off the rails for a couple of years, is now coming up due by the weekend, and sort of the Republicans and Democrats had come to a bipartisan agreement to just kick the can to March, but that was all thrown to the wayside. It looks like Elon Musk might now be running the country somehow, and decided it was time to throw that one out the window and start from scratch with less than 48 hours. So yeah, a lot of mixed signals, a lot of chaos. What can we do about it? I don't know, but what we can do is keep talking about the science, doing our best to drive a little closer to the truth.
Yeah, that's what we'll do. We're going to stay here in the U.S. and we're going to keep talking science.
Yeah.
So
absolutely.
help us do that. Les wants to know if you can ask a bird flu question. Sure.
Oh, of course.
Absolutely. Yeah. Shedrack from Nigeria, welcome.
Welcome.
Far away. That's cool.
Very cool.
John is in Southwest Florida.
Great.
Claire's in the UK. Welcome.
Fantastic. We've got a full roster of live streamers, even though we kind of mixed up our times this week.
People showed up. It's cool.
Glad everyone's here.
But Pete says he can't take Petey. I once went to a town at the north of Scotland named Tongue. Tongue. I'm sure many of you over there know it. And stayed in a little B&B, and they had a bar where they had just hundreds of single malts. We used to sample them. By the way, there's a place here in the city called the Landmark Inn. It's on the west side. I've been a few times. I haven't been there in ages, but they also have hundreds of single malts. And there's one night a week where it's 2 for 1.
Ooh, sounds pretty good.
Next time you come out here, Nels, we'll go there.
Let's do it. Let's put it on the menu. I was just trying to peek around the corner. I've got a bottle or two hiding out here in my office for both the celebrations and the rejections. It's good to have something stiff.
Dixon brought a bottle here for TWiEVO 1000.
Oh wow!
I don't remember what it is. There's only a finger or two left of it. People liked it a lot. All right, everybody's here. What's on the menu tonight, Nels?
Yeah, so this is fun. So we are cranking it up for the holidays. We've got a paper preprint that just emerged in the last month or so. And let's dive right in. So molecular and genetic characterization of sex-linked orange coat color in the domestic cat. This is a preprint on the bioarchive. Greg Barsh's lab. He's the senior author. Long history of contributions like this, getting to the genetic basis of coat color, sort of fur patterns in a number of animals, including this entry on the domestic cat. So we've got the Barsh Lab is in the mix here, headquartered for decades in the Department of Genetics at Stanford, with a footprint as well at Hudson Alpha Institute down in Huntsville, Alabama, and then some collaborators. So first author, Christopher Kalin, Kelly McGowan, and sort of some collaborators from Frederick, Maryland, so the National Laboratory for Cancer Research, hotbed of genomics contributions, I'm guessing, as well as Department of Neuroscience at Brown University in Providence, Rhode Island, and the Department of Anatomy, Physiology, and Pharmacology, College of Veterinary Medicine at Auburn University, also in Alabama.
So Nels,
Yeah.
let me ask you the fundamental question. What does this mean, sex-linked? What does that mean?
Yeah. So let's dive. So we got a lot of ground to cover. I'm gonna put that one off just for 1 minute.
All right. What's a cat? Tell us what a cat is.
Hopefully we're all up to speed on that one, and just sort of in the mood for thinking about the genetics of cat colors, as we're sort of hanging out around the holiday hearth, so to speak. So let's start with the evolution of these charismatic traits. So things like in domesticated species like cats, or we've talked about over the years on TWiEVO from pigeons all the way to corn to maize, the domestication of those species, and sort of then decoding the genetic basis of it and then putting it into that evolutionary perspective, which is on the menu for today. So TWiEVO 7 early days, Vincent, we were talking about entitled Pigeon Fashion Week Feathery Boots Edition. We had a fun interview with Mike Shapiro. So he and others have been taking advantage of all of the sort of selective breeding of pigeons for hundreds of years, which leads to these wild feathery boots, or on the legs, or the talons of pigeons. What am I going for here? All the way to checkered feather patterns, different varieties of feather arrangements, etc. We also talked about corn, maize a couple of times. I forgot about this one. TWiEVO number 30, Driving Miss Maisie, talks about genetic drive in corn, but getting more towards the domestication. We picked this up recently. Actually, last year's holiday edition, I was just looking before the show, TWiEVO 96, going bananas over the origins of corn. This was a really cool paper that came from Jeff Ross Ibarra's lab last year, and traces how corn or maize, or teosinte as a wild species, into maize, the domesticated one. And so we've talked about, so from kind of starting at the evolution, then we'll get back to sex-linked genetics in a moment. But so starting at the evolution, we think about natural selection is a pretty familiar or intuitive idea. The other phrase that's been used for these domesticated species is artificial selection, which is not really satisfying, because there's nothing artificial about this. This is humans sort of intervening. And in fact, Ross Ibarra, after our episode, we had a nice exchange on social media, and he mentioned that there's a commentary that Darwin actually thought about this, and instead of artificial selection, coined the terms conscious and unconscious selection, when sort of humans have a hand in the genetics or the breeding of certain species. That is great, actually. It's a really good upgrade from artificial selection. And so what we'll be talking about today with coat color of cats, orange cats, is clearly conscious selection, where humans thought these cats looked cool or charismatic with their orange fur. And so then the breeding has gone ever since, probably to the tune of 10,000 years plus or minus.
So if someone somewhere saw an orange cat, right, just spontaneously arose, and they liked it, so they decided to breed it. Okay.
Yeah. And so actually we'll get some hints about how, when that happened, or how many times that happened, kind of at the end here, I think, after we step through the genetics. So, turns out that coat color in cats is kind of a classic genetics puzzle, and actually had a hand in some of the early work, sort of foundational work in genetics, and thinking about actually, so sex-linked differences, X chromosome, Y chromosome, males, females. And we're starting to get to your question now, Vincent. So we're all familiar, I think, with Calico or tortoiseshell cats, which are this classic, held up, beautiful example of X-linked inactivation. So basically the females, there's genes or a gene or several on the X chromosome. If you're a female with 2 X chromosomes, one of those chromosomes is turned off, so that you compensate the dose of the gene so that everything, all your genetics kind of stays in balance. And that can happen during development or as part of developmental biology, as the offspring of a mother and father come online, where X inactivation is happening in different tissues at different places. And if the gene is turned on or off in these different places that relates to the coat color, then you'll get these patches, orange patches, black patches, white patches, that underline the Calico versus tortoiseshell.
All
so
right, here's some pictures of calicos, right? This very characteristic pattern, right? You got a white fur, and then you got black, and you got orange, and that's all because of random inactivation in different cell types you're talking about, right?
That's right, exactly. Those are clones of cells, which then are represented by those patches.
And then the other one is a tabby, right?
Yeah, so the orange cat, the tabby cat, there you go.
There's orange cats.
There you go. Yeah. And it can be, males or females can be all orange or tabby. So if it's a male, it just has one copy of the X chromosome from the mother. And so that's sort of the basis of the orange cats. And then the females would have to be homozygous for the gene that that relates to that orangeness, that trait of being orange. But if it's heterozygous, then you've got the calico, and sort of a similar scenario with the tortoise shells with slightly different biology. So all the way from genetics to popular culture, we've got famous orange cats. I'm thinking about Garfield, the cartoon cat. I'm thinking about Crookshanks from Harry Potter. The list goes on and on.
Why do people like cats so much, Nels? I don't get it.
Well, cats and dogs have a story of, in addition to, domestication, have really almost unlike any other species, ingratiated themselves to our own species, have really connect deeply in ways that are super impressive. I mean, these domestic cats and dogs have convinced us to feed them, pamper them, groom them, spend in some cases, half of our salaries towards their care. I mean, it's and guilty as charged, by the way. So I'm currently don't own any mammals, but have had cats in the past. Grew up with a orange tabby cat named Fluffy, which turned out to be sort of a mixed bag. Some neighbors had a litter of cats. We grabbed one, and I think it was sort of a half-baked idea, and I was too young to know what I was doing, and I don't think that ended well for certainly not the cat. I think we were all fine, but anyway. So how about you, Vincent? Are you have cats or dogs entered come to your door and gained?
When I was young, when I was growing up, and we had cats. I don't know why. And I went to college, so we had a couple of cats before college. And then I went to college. In my senior year, a cat walked into I was staying in a house with 3 other guys, and one day a cat walked in the door and stayed and became our cat. And then when everyone left, they said, "You take it, Vincent. None of us want it." So it was a black cat, and brought it home and lived with that for a while. Now we have a bunch of dogs. So I find, I've seen both, right? And dogs are very interesting because they really love humans. They are absolutely your best friend. No matter what you've done, they greet you when you come home, and they, really are happy to see you. And I don't get that. If you come home, the cat's not going to give a damn if you're home or not, right? I don't get this. They're very aloof, obviously, and that's perhaps what people like about them in part.
Right. Yeah.
And I know they purr. It's kind of cool that they purr and they sit in interesting ways. But Um, dogs are just. I never liked dogs, but they came into my life more recently, and I think they're quite interesting, especially small ones. I don't really like the big ones,
Mhm.
but they're just so um, happy about being with people. It's great, and
Yeah,
other people are not even happy about being with other people. So for a dog
good
to be,
point.
I think that's pretty cool.
For sure. I think they're both irresistible in different ways. Um, and so if it turns out, if we kind of loop back to the genetics, so Fluffy the cat was, if I think I'm remembering right, was a female cat and must have then been homozygous, had two copies of this gene called, that's been known or described for years, but not genetic basis hasn't been known until now. Sex-linked orange, SLO, is the sort of genetic region that's been identified. And so already it turns out there's all kinds known about, mostly from different species, about the basis of different colors of pigmentation, and going from sort of a dark pigmentation or eumelanin, which is sort of into the browns and the blacks, versus what we're looking at here. I think I've got it later in the notes somewhere. Um, pheomelanin, which
Mhm.
is the orange, and can be kind of combos of red and yellow. And by the way, we're talking about from freckles to feathers to fur.
Hmm.
And so a very common shared trait, but with different genetic basis. And this again starts to hint at some of the interesting evolution under the hood here.
So you can identify genes by just crossing animals and looking for the
Yeah,
propagation of the trait, right?
exactly. And so for a lot of those traits, they're known to be on autosomal genes. So they're not sex linked. They're on,
Mm.
23andMe, there's 23 chromosomes. They're on, like one of the 22 that are not the sex chromosomes that encode the genes that determine, whether the offspring are male or female.
Mm.
And so a sex-linked orange is sort of unique to the cats, or actually, I think they point out there's also a similar pattern in hamsters. So a sex-linked orange there, although probably independently got there, which is sort of an interesting, maybe future direction for the work, is how did the hamsters pull it? How did the orange hamsters pull it off? I think a little less irresistible than the cats and dogs. Their sort of level of success is pretty impressive, but not as sort of fundamental as cats and dogs. And my God, the video memes of cats, right? That's a whole nother
Oh
topic that we should
gosh,
probably
Instagram is just every other post is a cat video. Yeah.
yeah, exactly. It's a whole different ballgame. So anyway, X-linked activation. This goes back to the early 1960s when Mary
Mhm.
Lyons used sort of the calico observations of calico cats to sort of infer some of these patterns that were going on. And so, what's been known since about this specific sort of genetic region? So, maps to a single gene on the X chromosome, but that gene and the mutation or the variation that sort of leads to the orange trait, not known. Um, so we already mentioned, there's a ton of orange males because if you have one copy of the gene, the Y chromosome doesn't matter or can't for dosage doesn't matter. And so there's a lot of orange tabbies that are male. Females are homozygous, like my cat Fluffy from way back in the day.
You have to be
And so
homozygous to be female and have this trait.
correct, because remember so with the X inactivation, half of the gene is turned off, or half
Mm.
of the sort of gene dose is turned off on one of the X chromosomes. And
Yeah.
so if you turn one off, and if it's heterozygous, meaning you have the two different genes, then you might turn off the orange, and now you're looking at a calico pattern. But if they're both encoding for orange, you can turn off one, you're still orange. You turn off the other, you're still orange. It's an all orange female cat.
Orange all the way down.
All the way. That's right. Yeah. We're doing Mendelian genetics here. Um, so, um, so we already talked about, so some of the genes were already known. Some of the pathways are known. The most famous is one called MC1R. We're kind of an alphabet soup here, but this is melanocortin receptor 1, not on the X chromosome. So immediately you know that that's not the basis of orange for cats, whereas it is. And when we're thinking of some birds, maybe part of the story with freckles, even, et cetera. So, so what is this gene? And so before the authors kind of got into the game here, others had done sort of classic forward genetics where you map the trait. You take orange cats, calico cats, non-orange, I guess is the term for the two variants of the allele of the gene, and you breed them together. And then you ask, okay, who's orange and what's shared among them genetically? Who's not orange? non-orange and what's shared amongst them genetically. And so by doing that, they know they're on the X chromosome, so they can toss aside all of the other genes. They've narrowed it to a 10 megabase region. So megabases, we're talking about tons of DNA letters here, megabases of letters, and lots of genes, hundreds of genes really. And so still a lot of unknown or possible candidate genes that could encode the orangeness of cats. So first, what the group does here, the Barsh group, and by the way, it turns out I feel bad. So the bioRxiv, which the preprint server that houses all these preprints, was undergoing or migrating their files last night, and so I couldn't really get the preprint, even though it's freely available. It just wasn't loading. Broken link. So there's another preprint from another lab in Japan, Hiroyuki Sasaki's lab at Kyushu University, and they have a very similar study to this one, kind of complementary to it, but uses some of the same genomic comparisons, the same genetic tricks that we'll be stepping through, and comes to the same conclusion. They identified it kind of at the same time. This is one of the really fun things about cool science, Vincent, is that oftentimes there's multiple teams working on it, and they sort of show up at the same time with the same answers. I think in this case, the groups kind of knew that they were onto the same general story, decided to coordinate and post their work at the same time. And then presumably now both manuscripts are getting reviewed and will peer reviewed and will sort of show up at the same time. So neither group is scooped. It's sort of a tie, a scientific tie, and everyone wins, hopefully, in that scenario.
Yeah.
Anyway,
that's the best situation, right?
Yeah, absolutely. So what we're talking about the Barsh lab paper, but we could have almost as easily picked up the Sasaki lab paper, and I would have brought in at least parts of that to kind of compare. It's kind of fun to see how the similarities, but even some of the differences that still reach the same conclusions. But I honestly just couldn't get access to both papers and sort of in time. So anyway, so what this team does is then they narrow that down, that 10 megabase region, down to 1 megabase. So how did they do that? So we're in the genomic era, right? And so new genomes, people love to sequence stuff, and people love cats, and people, turns out, like sequence cats. And this is like big industry in many ways. So Purina, for example, that sells a lot of cat food, is sequencing cat genomes. There's a cat reference genome. Other people, the breeders, will sequence the different breeds to get a sense of what makes their breed so special. The Sphinx cats, the tabbies, sort of the fancy ones that you see at the cat shows. And so using that information, they actually were able to narrow this down tenfold by mapping. So what parts of the genome are common to the orange or common to the non-orange, but not overlapping in a sense. But then, from there, they actually, so by narrowing down to a megabase, they then could identify 50 mutations over that sort of 1 megabase region. And they're taking a liberal definition of mutation. So these are point mutations where the DNA letter changes, but they're also duplications of sequences that could be, duplicating 1,000 letters, 1,000 base pairs, deletions, insertions of transposable elements. These are the selfish genes that hop around. They act as mutators that can kind of diversify, add variation to genomic regions. And so they are able to identify sort of 50 changes or mutations through different mechanisms that are sort of candidates in that region.
Okay. This is compared to the non-orange cats, right?
Well, no. So actually right now, this is just compared to the reference genome, which was not an orange cat. So it's just comparing it to one non-orange. So but then what they do is actually they can go from 50 mutations down to 3 by doing what you're saying. So all of these, cat breeders, et cetera, when they look at those genomes and say, okay, well, this breed is orange, but this one's not. And how many of those 50 changes are in both? 48 of them are. And so cool. So I think they had 51, I might have the numbers slightly wrong here, down to 3 candidate genes. So one of the differences that's like now all oranges have these 3 differences from non-oranges is a single mutation in one of those retrotransposable elements, a sign element. We had a nice run, this might be a couple of years back now, Vincent, of papers where we were looking at, cool traits, sort of charismatic traits, where it was transposable element sort of hopped in and changed or it caused variation that led to a cool trait. I think moths, we talked about the peppered moths, right?
Yeah. Yep.
Some butterflies, and again it was transposable elements. So that's kind of like, you see that and you're like, ooh, cool. This could be something. The next one was a repeat sequence that had expanded to some degree. And then the third candidate was a 5 kilobase, so 5,000 DNA letters deleted near a gene called, this is alphabet soup, ARGHGAP, RGAP 36 is the gene. Okay, so they could kind of actually prioritize just based on sort of what's known about these genes. So RGAP 36 is pretty interesting to them because it's involved in some of the cell biology that leads to whether a cell is pigmented or not. So cells, there's pigment, which is like a physical thing, dark, orange, whatever color it is, gets moved around in certain cells and compartments, and then added basically to a hair follicle in the development of like a hair fiber. And we won't dive deep into that at the moment, but suffice it to say, this is one of the kind of, parts of the machine that moves the pigment around to get into the hair follicles, and then ultimately gives you that fur, whether it's, out on the surface of the cat. So that was one that I think really kind of narrowed their focus. And so they sort of chased that one. So RGAP, this is short for Rho GTPase activating protein 36. It's involved in membrane, a lot of the family of genes, the Rho GTPases are involved in membrane trafficking. So moving sort of molecules from compartment to compartment within cells. Lots of links to the cell biology of pigmentation. Ironically, this gene doesn't work this way, and we'll get to that sort of at the end here. But it was one that, or a function that sort of, I think, made it easy to prioritize that one. So what do they do next? They look at more orange cats, the genomes of more orange cats, and say, okay, does this deletion map to even more examples, independent examples of cats being orange? And the answer is yes. So I think they looked at 150 more orange cat genomes. They all had that 5 kilobase deletion near this RGAP 36 gene. They looked at about 50 non-orange cats, and they all lacked that deletion. So guilt by association, right? This is looking pretty good. And by the way, that's not the case for the other 2 candidate regions. Those, in some cases, those don't correlate perfectly with coat color. So it's looking good. And then the question becomes, well, what is this RGAP 36 doing? How is this, in this deletion in particular, near the gene? So that deletion is not in the gene, but nearby. And so immediately when you kind of, as a geneticist, when you see that, sort of scratch your head and you say, okay, well, this probably is regulating the expression of that gene. You're not mutating anything in the body of that gene that encodes the protein that would change the amino acids, but the amount, the dosage of that gene into protein is sort of a logical way forward. So they look at the expression of RGAP 36 in skin biopsies from cats. And, thinking about the cat industrial complex, you can get this tissue pretty easily from a lot of shelters or veterinarians, et cetera. And so they look at, they get orange cats, non-orange cats, and then they can measure how much of that gene is turned on in the skin tissue. These are sort of early neonatal skin samples. How much of this gene, RGAP 36, is turned on in orange versus non-orange. And in orange, it's up 13 times over non-orange. That's pretty drastic, right? So, and it's a little bit paradoxical, actually. So we think about deleting regulatory sequences that sort of, influence the expression of something. Usually we'd think it would turn down the expression. Here it's the reverse. It turns it up, which is sort of an interesting little genetic curveball there. Okay, so that's cool. And there's 6 other genes in the neighborhood that are not changing at all, whether they have that deletion or not, whether the skin biopsy is from an orange cat or a non-orange cat. And this is important because, so regulatory regions near genes, the genes that actually encode the proteins, it can get complicated. And so just because the closest gene in the neighborhood is RGAP 36 doesn't necessarily mean that sort of regulatory region is controlling the expression of that one. It could be sort of jumping, leapfrogging a couple genes over and controlling that expression. So they dotted their I's and crossed their T's. They looked at those other genes and don't see a difference. So again, they're doing this really nice kind of, almost slow march to figuring out the genetic basis of the orange coats of cats, of tabby cats. Okay, so then there's a lot known. You start to kind of merge these lanes, the genetic discovery and the follow-up, with what's already known about the pathways that are involved in getting the pigmentation out into the hair fibers. And so MCL1R, we mentioned this receptor, which is kind of central in the movement of pigment. They can start to look at the expression of that gene itself that encodes that receptor, and others that are sort of have been put into complexes that signal the cell biology for pigmentation. And in fact, they see in the orange cat tissues, skin tissues, there's reduced expression of those other ones. So you upregulate RGAP36, and that looks like it's coincidental with the downregulation of MCR1 and some of these other genes. And that actually, so we're kind of going up and down here. We're pulling the strings on the pigmentation biology, but that kind of makes sense. So if you downregulate MC1R and these other parts of the pathway, you get less pigment out to the hair fibers. It's not as dark. It's not that melanin anymore. It's the sort of the other flavor, the red, orange, and yellows instead of the more browns, grays, and blacks. Okay, so I'd say, so far so good. It seems like they're marching on the right trail. There's another lab, the Sasaki lab in Japan. They're looking at the genomes of feral cats and pet cats from Japan, seeing this. They're kind of marching into the same pathway here and seeing the same, narrowing in on the same gene. So then they gear up to do some functional studies, which I think really kind of put the nail in the coffin of identifying the right gene here. So they get more samples from other sort of neonatal cat samples, and do in situ hybridization. So they can make a probe to the RGAP36 gene sequence, and then see whether they see where is this gene turned, not just is it turned on or turned up 13x, but where is it turned on as it relates to the skin biopsies. So they actually have a really beautiful figure where you can see the probe is lighting up right at the base of the hair follicles, and for the orange cats, and not so for the non-orange cats. And so the gene, or the protein encoded by the gene is at the right place, sort of at the right time, and missing in the non-orange cats.
And this regulation of the gene is not seen in any other tissue.
Yeah, right. Oh, good point. So it gets complicated. So in most genetics is, right? So it turns out this gene, SLO or RGAP36, actually you do find it in other tissues. So it's in a lot of neuroendocrine. So it's involved, again, in this sort of signaling, involved in moving membrane trafficking, moving around compartments that contain cargo. So it's actually in some brain tissue, some tissue from glands, but there's different isoforms of it, and some are expressed in some areas and not. It looks like the 5 kilobase deletion specifically influenced the expression in these, in and around the skin tissue, but not in those other tissues. And that's probably important here, because those other tissues where this gene is more stable or more expressed, it's essential. And so if you mess with it, if mutations show up that mess with that gene in those tissues, it's not a viable cat. It's not going to make it. And so that kind of, it's an important point, Vincent, also because I think it illustrates how kind of rare, it's like a jackpot. It's like winning the lottery. If you're a cat breeder, to come up with this 5 kilobase deletion that gives you that orange trait. Really hard to pull off.
So you're saying it's not increased in other tissues, because that would be lethal.
Correct. Yeah, exactly.
So whatever this regulatory sequence deletion is, that's the effect of it to be tissue specific, to be specific for the skin, right?
Yeah, exactly.
Interesting.
Which is sort of, yeah, exactly. And it's also, it's that paradox where you delete it and then it's upregulated. So it probably, or they, it's speculating, but the authors say that they think it might, sort of by splicing that sequence that was 5 kb away, 5,000 letters away, actually uncovered an enhancer element, which actually turns up the expression as opposed to turning down.
It could be. Yeah. It could be specific for skin and not other tissues.
Yeah, agreed. Or all of the above. Yeah, it could be multiple things going on there, but it's ends up being one of these. I mean, this is what's great about evolution, right? So you have all these rare events, but if that are random, but if selection acts, in this case, conscious selection, cat breeders, there's probably some rare small number of orange cats in ancient times, but humans said, "Hey, that's really cool. I want more orange cats. Let's keep these things. Let's breed them." And that's the origin. That's the origin of orange. On the origins of orange. Okay, so they go on, Vincent, and there's all these great, genome. They've depended on the fact that there are people sequencing the genomes of a lot of cats to do these incisive comparisons. But there's also, so they do a different flavor of genome sequencing, single cell sequencing of some of these orange skin tissue samples. And so now they can look at the level of genes, not just this gene, SLO or RGAP 36, but they can look at all the genes, and specifically the ones in the hair follicular cells, or the sort of the cells that they care about. And what they see is there's actually not just the sort of 6 genes that they've considered so far, but there's 450 genes that are differentially expressed when they compare orange versus non-orange tissues. And so some of those genes are probably just kind of, hitchhikers, or they're just coincidentally up or down. But others, like in the MC1R pathway, are ones that are, explaining what's going on, presumably with the pigmentation, the reduced expression of those ones that lead to the orange. So the very last thing they do is try to get at a little closer to the mechanism. So what is it that having more of this RGAP 36 in the game, specifically in these cells, is doing to the signaling pathway? And so this is where there's kind of another curveball. So RGAP 36, one of these road GTPases, is generally involved in sort of facilitating the membrane traffic. But it needs, in order to do that, there are these sort of GTPase cycles that we won't go into the weeds here too much. But RGAP 36 has a problem. It has a mutation, so it doesn't have the catalytic ability to cycle GTP. Instead, it looks like it interacts with another protein kinase A, in a way that short circuits the signaling that usually goes on to promote the eumelanin, the dark pigmentation. And so it sort of puts up a roadblock, in a sense, a cellular roadblock, and says, okay, signaling isn't happening. It sort of short circuits that message. And so then you don't turn on all those genes, which would bring the dark pigments, and you end up with an orange cat.
Cool. That's
pretty cool. Yeah. So they've gone from kind of that, sequence gazing the genome all the way, drilled down all the way to they think they know how it works. And, it's kind of cute when it relates to orange coat. Yeah. But scientists like you and me get kind of excited about this, because it's like, oh yeah, now we also know something more about this signaling pathway. We know something more about this cell biology, which is also going on in other tissues. The output might not be like as charismatic as the orange coat, but it might be a key to hormonal signaling or how our brains work. And so there's, it's sort of the same modules that kind of show up again and again. And the orange coat of the cat just gave us a cool sort of wedge point in to learn how some of this biology works.
Yeah, I was going to ask you, like, what's the value, besides an intellectual satisfaction, what's the value? And yeah, so you can learn about other pathways too, right?
Yeah. And so this hints at the idea that geneticists really, they're kind of connoisseurs for the phenotype. What is the trait? And if it's something you can measure, you can use that to then say, okay, what maps with what correlates with what, and that sort of drives you. So some of the other traits associated with this are a lot harder to measure. Did you secrete a certain amount of a hormone to keep the adrenal glands working? Probably more essential biologically for sure, but a lot harder to measure. And so this is sort of a fun way to get a view that would otherwise be hard to see, and then apply some of those ideas. You can sort of falsify or test those ideas in those other tissue types or those other sort of biological processes.
I suppose there's not a shortage of funding to work on cat biology.
Well, yeah. I mean, at the top, we talked a little bit, we could be about 24 hours away from the government shutting down for a while, who knows how long. And so, we might be like hitting up Perina for some more research funding pretty soon here. Private, public, hybrids, diversified portfolio of research funding.
Let's see. Looking for their funding sources here.
Oh, yeah, good question.
It's research has been supported by NIH.
Okay, there you go.
And the Hudson Alpha Institute for Biotechnology. It's
Public,
interesting.
private.
NIH, it's about human health. So you have to pitch it that we're going to learn about human health somehow, right?
Yep. And that's right there, just as we were kind of talking about in the last minute or two. So, but Vincent, our podcast is not This Week in Cats, but it's This Week in Evolution. Yeah. And so the other cool thing that I can't help but kind of linger on is that, so now that they know this gene, now the evolutionist in us can kind of get excited, because with that gene in hand and the deletion nearby that gene in RGAP36, you can use that to very precisely compare this to other cats. And so, we already did the comparisons of orange and non-orange, but the evolutionist wants to ask, okay, is RGAP36 in play in other species? Is it part of the orange hamster story? Other orange creatures out there? And the answer might be maybe. The other thing you can do is ask, how long ago did that mutation arise, right? And so it's not in this paper, but someone was commenting on the work and noted that there's known to be some mummified Egyptian cats that were orange. And so assuming that some of those, that the skin tissue of a mummified cat is preserved and somehow available, you could use some of these genetic tricks and some of the technology that's been put towards ancient genomes and ask, do we see that deletion, that 5 kilobase deletion near RGAP36 in mummified cats? That might get us to the origin of orange, where it was those cats, or right around that time, or the near ancestors when that came up. Or it might tell us, no, this was more recent, that maybe those orange cats from the mummy, the Egyptian days had a different origin.
I wonder how they know in a mummified cat that it had orange fur. I mean, usually the coloration is gone. But, many museums collect specimens like this, right? And they can go back some years. I the koalas in Australia that got infected with the retrovirus, they went back, they had some museum collections of skins, and they could tell that it was there, 150 years ago already. So maybe you could do the same thing in cats. But you said at the beginning, how old are these orange cats? Do we know?
I don't think we know. So that's like I didn't do a deep dive into that, but it sounds like, it could be that far back. And you're right. I don't know that the fur is actually preserved. The skin tissue, hopefully. Yeah,
the skin would be. You could look in the skin, but you wouldn't know. You'll find out afterwards if they're orange or not, right?
That's right, based on whether you see the deletion or not. And that might be cobbling together other parts of the historic record, right? So, for example, are there hieroglyphics that sort of, it's like the cat next to the symbol for, or I don't know, this is being a little glib, but there's other evidence. And so building that case from both a historic and genetic record is actually possible. And we've done some studies like that along the way too, thinking about the preservation of infectious microbes from the remains of humans, et cetera. There's all kinds. That's sort of a really cool growing field in evolutionary genetics.
So this is not of real value for cat breeding, right? Because you just breed them and you get orange cats, right?
That's right. So we are. So this is a great point about sort of applying genetic technology versus the old-fashioned way. Probably not for this trait because it's so easy, but if you believe me, there are, I'm sure there are breeders out there or collectors out there that it might be that orange is not part of the sort of genetic heritage of that breed. And so
you could introduce it, right?
You could introduce it. You could, and using CRISPR sort of technology, you could take a run at deleting something like that 5 kb region, and all of a sudden bringing orange coats to a breed that might have never had them naturally. It also, I think this is on the, and it's a serious business for sure, collecting and showing cats, for example. But it also, it's a similar thinking that goes into like the domestication of crops, right? And so In that case, cats as well, the generation times are pretty long. And so how long it takes to do the sort of old fashioned breeding versus kind of the scalpel of genetic engineering to bring in favorable traits, things like historically pesticide resistance, or, there's now strains of rice that have more nutrition engineered into them, or pest resistance, all kinds of possibilities, favorable traits. Of course, that's got caught up in a political conversation. GMOs, genetically modified organisms, has become a sort of morass, and and there are some real ethical considerations for sure, whether it's cats, crops, or for sure humans. But the backlash in a lot of cases is pretty severe, and I think we're suffering some of the trade-offs of not pursuing some of these things, especially I would say
most of the world's rice is GMO, right?
Right.
And so it really helps a lot. So
yeah, I think I even saw, non-GMO has become such a buzzword, and I even saw advertised non-GMO salt.
Yeah, I was going to say, I've seen that too.
Yeah. So we've kind of lost the script, or lost the plot, as they say in some of these cases. Yeah.
So we have some comments here that
good.
We'd like to highlight. Yeah. Jason said, "Cats are the creatures who run the internet."
That's for sure.
When they get bored, it goes down, right?
Yeah.
Tom says, "Charismatic traits," a great term. So what else would be a charismatic trait? Anything that makes an animal more appealing, like a dog or a cat, I suppose, right?
Oh yeah. So for the pigeons,
the pigeons, yeah, that's right.
These feathery boots that were the breeders like they see, that's a charismatic trait. That's a fancy pigeon right out of the gates. Chickens, there's some charismatic traits there.
I think probably horses too.
Oh yeah. For sure. Yeah. Thoroughbred horsing. These are charismatic creatures, charismatic traits. Again, plants, plant breeders are pretty particular about, think about orchid. I was just at a local orchid show here, Vincent, in Salt Lake a couple months back. And oh my goodness, the variety, quality, the beauty, the inspiration of these orchids as they were flowering in this sort of high concentration, it was really something. That's, humans have really taken these charismatic traits to the next level in a lot of cases.
Barb Mack says, "Cats convince you you're the best. Then they treat you like a slave, but you love them despite that, because by then you're hooked." I see all these videos on Instagram of cats like jumping in Christmas trees and knocking them over, jumping in aquaria, right? I mean, what is that kind of behavior? I don't get it. Why do they have to jump on it? I even saw this one where they hung the Christmas tree from the ceiling so the cat couldn't get it, and still the cat climbed on something and jumped onto it.
They're the best. They're unique in their ability to grab our attention and touch our hearts.
Elizabeth says, "I have cats that run up to me when I come home. I have one that even wags his tail." Well, that's right. I'm sure that my experience is not necessarily the norm. Yeah. Marcus here from Helsinki, 3 degrees. Welcome.
Welcome. Good Yule. Yeah.
John is doing something in the snow while listening. Welcome.
Minneapolis. Fantastic.
Yep. Ankley is in Norfolk, England. And Kang is from Chicago.
Welcome.
There was a little discussion about feline immunodeficiency virus, but I think others have figured that out. Then here's a vet in Oregon who's listening in.
Great.
Among veterinarians and probably cat enthusiasts, there's common experience that orange tabby males have color-linked personality. Orange males tend to be more dog-like.
There you go.
They are food-motivated, more social, interactive, teddy bear-like cats. They might even convince Vincent that cats get excited by you coming home and are a good companion.
There you go. Huh. I'm sure there is something to that. There is a genetic story under the surface, a linked story under the surface there. And, the genetics is going to be a lot more complex. But again, that correlation, which is probably not perfect, and it's because it's more complex, but is a pathway forward for research of the future to use that opening of the charismatic trait, that orange trait, to then understand something about the behavior. in behavioral genetics, oh my God, like this is obvious, it's an obvious hotbed of interest with, honestly, pretty modest progress, because getting at these more complex behaviors that might not be as easy to measure as whether a cat's coat is orange or not, the degree of difficulty goes up almost exponentially. And so this is kind of a foothold, I think, to inspire future research along these exact lines. Yeah, and thank you for the backstory there on the orange tabbies.
So Tom says, near as I can tell, lots of cats are food motivated.
For sure. We're in good company.
Thomas, thank you for your support of science communication. We really appreciate your confidence in us.
Awesome.
And Thomas has a question. Talking about GMO crops, I can't believe how different wild banana species are from the selectively bred and genetically modified varieties. Yeah.
Thomas, agreed. And so, Vincent, we did not quite as deep of a dive as we did on the maize, the domestication of teosinte into maize, but we did so the reason last year's holiday episode is entitled Going Bananas Over Corn is that there was a domestication of bananas paper that came out right at the same time. And you're right, Thomas, the wild species varieties are really something. I would also hold up the potato as all of these sort of the variation in potatoes, sizes, shapes, colors, textures is another one of these examples that is, really, cool. Really interesting. Yep.
What was the other? Okay. Here's from Kang. Is the splotchy coloring that in some cats genetically regulated by region from head to tail?
Yeah, it can be. So we kind of glossed over. So there's the exon activation going on, but there can actually be in some cat breeds, so I think it's very common to see different pigmentation or color kind of on the extremities, whether it's the tips of the ears, the tips of the paws. And it turns out there's temperature regulation underlying that's or sort of merging with some of the genetic regulation. And that leads to differences in pigmentation. These are the Siamese cats, I think, or some of those breeds that are something, the body is mainly white and then it gets more gray or dark as you go out to the extremities. And there's some really cool genetics meeting during development, meeting with environment.
When I was a sophomore in college, I took genetics with Jerry Fink.
Oh, yeah.
And one lecture, he came in with 2 rabbit pelts he put on the counter. One was white and one was brown. He said these are both genetically identical. They were just raised at different temperatures.
Yeah. There's a great geneticist up in Montana, a guy called Jeff Good, who's gotten some of the genetic basis of coat color difference in snow hares, seasonal differences along the lines that you're talking about. Jerry Fink, that's a great name, sort of historic figure, yeast genetics. And
so I had him in 1970, 1972 or something like that. He was at Cornell, and then years later he became the head of the Whitehead at MIT.
That's right. Yeah.
And I remember seeing him walking around, and I went up to him. I said, "I took your genetics course not too long." He said, "Yeah, I guess we both survived."
Yeah, that sounds
very cool. Yep. Barb Mack says she can't put up a Christmas tree because her cat is a jumper and would have it down in seconds. Yeah. So what is this? Why do they have to take it down? What's the motivation for that?
So I don't know about the trees, but I do know, or I saw something. I think I didn't read it closely, but all the scratching, on couches and whatnot, there's a deeper meaning to that. I think the cats are trying to communicate or engage through some of these behaviors. I don't know, the tree, that one's that's beyond my pay. That's above my pay grade.
So Nels, when I was in college, this cat who I ended up owning had one red eye and one green eye. What's the genetic basis for that? Do you know?
Oof, good question. I'm not sure on that one. Eye color genetics, I don't think is as straightforward like these things. So it's maybe a good opportunity. So some traits, like as I was trying to make a good case for why these, visual traits can be really good footholds for genetics. But I think in some cases, even the things like human eye color, I might be wrong about this, but I think it's more complicated, if you'll forgive me, than meets the eye. And so the genetic basis of that is when it's complex, even sort of simple traits that are easily measured or are not necessarily easy to dissect the genetic basis of the contributions.
Yeah, Sean says it's chimerism.
Oh, on the eyes?
Yeah, I guess. Yeah.
Yeah, makes sense. I mean, that's what's going on. So that would, in many cases, that implies sort of X-linked contributions, sex-linked contributions, as is the case with sex-linked orange in the cats.
Elizabeth says they aren't trying to take the tree down; they're trying to climb it. Climbing trees is okay. And Jason says the scratching is to sharpen their claws. Okay.
Yeah.
Rob, thank you for your contribution to science communication. Really
fantastic. Yeah.
appreciate your support and confidence in our ability to do this. Okay, we want to do some picks, Nels?
Okay, Vincent, I've got one that late breaker that really hits close to home. So about an hour or two before we were coming online for the live stream, one of my colleagues stepped in and said, "Hey, have you seen this new thing from Google called Notebook LM for, I think, language model? It's geared towards scientists or science curious folks. And this is artificial intelligence taking a stab at science podcasting. We might be out of a job here shortly, Vincent." So the Google Notebook, you give it a PDF file and then click a button for a audio overview, which is basically a science podcast. And a couple minutes later, you've got an audio file of two, robot voices, or it sounds like real people, podcasting about that paper. So what I did, Vincent, I'm going to, you know where I'm going with this. I'm going to queue up here. I took the PDF for this preprint. It did a short, title, or summarize the title, Genetic Basis of Orange Coat Color in Domestic Cats. I guess that's the title of the podcast. I think our title's better, On the Origins of Orange. But then it produced an audio file. So I'm going to press play here, and we can listen to what Google, we'll just do about a minute, what Google thinks of this paper.
Welcome back, everyone, for another deep dive. This time we're going to be looking at those beautiful ginger cats and the genetics behind their vibrant orange fur. We'll be exploring excerpts from a paper called Molecular and Genetic Characterization of Sex-Linked Orange Coat Color in the Domestic Cat. So before we dive into the nitty-gritty science of it all, let's just set the scene a bit. I'm sure you've all seen Calico and tortoiseshell cats with that amazing patchwork of orange, black, and white fur. But did you know that their unique coats are actually a visual representation of X chromosome inactivation?
Yes, that's right. It's a phenomenon that happens in all female mammals. They have two X chromosomes, right? And during development, one of those X chromosomes in each cell is randomly switched off or inactivated. So in a calico or tortoiseshell cat, each patch of color actually shows you which X chromosome is active in those cells. That's how they get that kind of mosaic look.
It's like each patch of fur is telling a little genetic story.
It's like each patch of fur, Vincent, is telling a little genetic story.
That wasn't so bad, actually.
Well, I know.
He got the guy even going,
Exactly.
which really bothers me that they programmed that in to have him say randomly, okay, so they don't wander off onto different topics, okay? They just stick to the paper and that's it. And I think that's part of the beauty of a podcast is that you get a bunch of people together and anyway, so they can talk about the paper, but you and I can draw on our years of
that's right.
So I can pull stories out of the thin air that I had this cat with two color, and the computer can't.
I'm with you.
What I'm saying is basically podcasts by people have personality, and that one's going to sound the same every time. It's done, right? It's going to sound exactly the same.
Possibly. So it's, so they're not programming it, it's training. So actually, I wouldn't be surprised, Vincent, if there are snippets, some phrases that it's grabbing from you, because all of your public online podcasts,
that's what they use for that. Yeah.
Are there. And so,
interesting.
So it has that veneer because it's drawing from us. of our conversations and sort of echoing it back with sort of a veneer of that. But I agree with you. I don't think so. For better or for worse, we're now north of an hour of talking together because we just can't resist sort of these deep dives and the meandering, the rambling. That audio overview or AI podcast was about 11 minutes. I haven't listened to the whole thing, just that first minute or so. So I'm still, actually there's a cool beta test. You can do an interactive mode. So I'm thinking about, getting some of my students or some coming across grad students, postdocs, et cetera, who might have some curiosity of podcasting, maybe to show them this, of trying the interactive, like trying your own podcast where you're interacting with the AI. So you're sort of sharpening up your own conversational skills in that podcasting space. It took us about 9 years on TWiEVO to get to where we are today. Or for me, certainly, you've been doing this for a while longer. But but yeah, I agree. I'm still holding out for the meandering and the rambling, that sort of personal contact or personal experiences that we're weaving in, is that there's value in that. I think there's value in this other thing, but it's different.
So someone has said to me that if you wanted a summary of a paper, you could do that, right? You throw the paper onto it, and they summarize it for you with some background. To which I said, well, you could read the abstract and maybe the introduction. That would do the same thing. But fine, if you want.
But this is a great tool. So just setting aside the sort of podcasting mimic for a moment, that's what it's trying to take that abstract, but put it into a less sort of technical or professional sort of phraseology. And that could be worth a lot, especially if you're, in grad school, if you're just science curious, a way of sort of accessing literature. Now, hold on though. There's one thing, trust, right? So we're bringing to bear our decades of training in virology, genetics, evolutionary genetics, whatever it is, to try to make sense of the work, to try to decode it. Yeah, we're promoting it. We're trying to kind of put it in a positive light for sure, but we're hoping that and we get it wrong sometimes, maybe on the details, but what we're hoping is that, from that big picture or the kind of conceptual idea, that we're pointing the audience in the right direction. AI hasn't cracked that yet, right? So it can really pull you off the rails with a lot of confidence, but maybe not a lot of experience or truth behind the information that it's trying to summarize or express. And so I think I'm not ready to hang up the shoes yet for TWiEVO. We'll be back for episode 110 and on.
I think listeners like people, with all the things that go with people, the good and the bad and everything. It's, computers can't do it. Maybe not yet. Maybe they will at some point. But just people. You and I are folks with our foibles, and that's what people like to listen to, I think, right? So I think it's amazing. We have some amazing reactions here. Bartback says, I don't know whether to be impressed or horrified.
Isn't that something?
Harry Ozone says, yikes.
Really
Jason says, people are going to get really quick at spotting this faux voicing.
Yeah. Although that's, like you're pointing out, I mean, immediately that the same thing struck me, Vincent, which is the pauses, the kind of that I'm about to read the title moment where, I mean, I've faced that probably on this podcast. If we rewind the tape, I probably did the same thing.
Oh yeah. We do it all the time. And Tom says, oh, great snippets grabbed from you. Yeah, they will. They can do that if they're smart, right? Jason says, all of it, AI training has serious elements of unidentified masked kleptocracy.
Yep.
That's really good.
There are, putting stuff out in the public domain it has some trade-offs. The tragedy of the commons to some degree.
But the AI's tone has a weird, inappropriate, jolly quality. Maybe they also trained on AM radio.
That's possible.
They probably they program it with some AM radio clips, right? With that, hey, listen to the top 10.
I think it's grabbing from a lot of sources. Yeah.
Jamie says, entertaining, but no match for your real human talent. Nels is right. Could be a good training tool.
Yeah, good way to kick. I'm kind of tempted, or if one of us can't make the next one, maybe you can try. If I can't make the next one, you can try your hand on AI me
Well, Jason said, I'd like to hear TWiEVO when you pretend to be AI bots.
There you go. There's another way to do it.
Nels,
Right.
and the tone reminds me of Crash Course.
Oh, I don't know if I know that.
I don't know what it is either.
Another AI situation.
Thomas says,
Great.
I couldn't use an AI podcast. I need the human quirks.
Yeah. Yep. I mean, we're now,
But, you know,
an hour longer than that summary, but I think we
all
probably.
Sorry, go ahead.
Well, I was just going to say, I think we've covered some great additional ground.
Yeah.
That is I mean, there is I can't remember who said it, or it's probably multiple people, but the notion that we're, if we outsource our writing or our poetry or our term papers to AI, we're sort of missing the point of what we do as humans. And so if AI is, more efficiently washing our clothes or doing sort of those tasks and freeing up more time for us to podcast or write poetry or whatever, like that seems to me like a good use of the technology.
Yeah, for sure.
Yeah.
Well, I do have a pick. It's a not as interesting as Nels by far.
I got lucky on this one. It's a good year-end science pick. What's yours, Vincent?
It's a Nature article by Gemma Conroy. Humans evolved for distance running, but ancestor Lucy didn't go far or fast. So this is a really interesting article. It's based on a current biology article. Lucy is Australopithecus afarensis. So this is a small hominin that lived over 3 million years ago, right? And it walked on 2 legs, right? And they find footprints of it, and they can figure out how fast it could walk, right? But and in fact, this particular species is a favorite for people figuring out how bipedalism evolved, right?
Yeah,
but
that's right.
Humans are thought to have predominated because they could run, right? They could outrun a lot of species for longer periods of time. They have endurance. They can run away from animals chasing them, trying to eat them.
Predator prey.
They have this group built 3D models, which try and estimate how fast it could run, right? And they show that, they look at muscle mass and length and tendon length and bones and so forth. They estimate that Lucy could reach only 5 meters per second, whereas humans can go 8 meters per second. So it was a bit slow. But I think this is very interesting, the evolution of running, right?
Oh, yeah, for sure.
So the original article is in Current Biology. Of course, this is paywalled because it's Nature, but you can go check out. Maybe the Current Biology one is open. I'm fascinated by these stories of studying human ancestors. And I think it's really interesting what you can find.
Super cool. There's also one that just came out, big splash on interbreeding between what's now modern humans and Neanderthals. A bigger revision on the dating of that based on some really cool work from a couple labs.
I had a pick on TWiEVO recently where they found 2 different species of hominins footprints not too far from each other. So they coexisted like 1.5 million years ago. Maybe they even saw each other walking, and so we never know who's around with whom, right? We just know there's parallel evolution, but the footprints were clearly of both species. So it makes you wonder, like, did they wave to each other? Did they try and chase each other? What did they do?
More than that.
Really fascinating. Really, really fascinating.
Yeah,
right.
And
then
then
they attack each other, right?
Yeah. And then seeing the sort of fossil record, archaeologic record, like in this case, but then as that merges with the genomic record, I think that's I agree.
Yes, very
really
much.
a cool area also of sort of a growth industry in human genetics and in evolutionary genetics as well. I think it was Priya Mirjani's lab who is one of that really spearheaded this genomic dive into integration with Neanderthals. Really inspiring work.
Let's see here.
I could, Vincent, I could play another minute of that AI podcast.
No. I'm looking at this comment here. So Jason writes, parametric meta pattern recognition Stephen Wolfman recently pointed out that the first obvious difference is that AI LLMs give different result answers each time. He found that very useful.
That's interesting. Yeah, these large language models, right?
Better get used to running again if they decide to bring back the saber-toothed cat along with the mammoth. Yeah.
We're mixing together our picks and our papers. I love it.
Yeah. All right, that's a TWiEVO 109. It's the last for 2024. Hope you enjoyed this year. We hope you have a great holiday. You can find the show notes at microbe.tv/TWiEVO. If you like what we do, consider supporting us, microbe.tv/contribute. Nels Eldy's at selvolution.org and over on Blue Sky, L. Earlybird. Thanks, Nels.
Yeah, thank you, Vincent. Thank you, everyone, for being here and all the support so far along the way up till 2024, and looking forward to connecting in the new year.
Thanks to our moderators, Barb Mack, Tom, and Les, and everyone who joined us today to listen on in. I'm Vincent Racaniello. You can find me at microbe.tv. Music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. It's too loud. Let me lower it. But by the time I do, you've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious. Thanks, everybody. Bye-bye.